I would like to welcome you all to the webinar From Gene to Function to Therapy with Dr. Michael Dean today. I'm Christine Vokey, Director of Education and Information at PXE International. I want to tell you a little bit about Dr. Dean. He's the Deputy Director of the Cancer and Inflammation Program at the National Cancer Institute in Frederick, Maryland. He's also the Chief of the Human Genetics section there. Dr. Dean obtained his PhD from the Biochemistry Department at the Boston University School of Medicine, and he performed his postdoctoral studies at the National Cancer Institute on the MET oncogene and the cystic fibrosis gene. Of interest to PXEers, Dr. Dean has been participating in research for a number of years. For the last 8 years, he has hosted and organized the annual North American ABC meeting, which is a day-and-a-half meeting dedicated to discussion and presentation of data from graduate students, postdocs, and senior investigators, all studying the role of ABC proteins in human disease. For several years now, an entire workshop session has been devoted to PXE. The 9th annual North American meeting should be taking place in 2012. And with that, I'd like to welcome Dr. Michael Dean. Thank you for being here, and you can take it over.
Okay, thank you very much, Christine. Welcome everyone to this webinar where I'm going to give an overview sort of in general how we approach genetic diseases in terms of finding the gene and going from there to understanding the function of the gene in both the normal condition and also in relation to the disease. And then hopefully from that move to therapy. And I'll provide some examples relevant to PXE along the way. Okay, this is a diagram of what all of the cells inside of our body look like. We have billions of cells inside our body, and they have, let me get the little dot back here. They have all this basic structure. There's a membrane on the outside of the cell, which basically holds everything in there. Inside the center of the cell is the nucleus where the DNA is, all the genetic instructions to make all the proteins for our body. And then shown out here are proteins being transported out to different organelles. The mitochondria is a special organelle that generates all the energy of the cell. There's a whole secretory system where proteins that are getting secreted, like for example insulin, get processed and then moved outside of the cell. And then these arrows here show transport because it's becoming appreciated that the transport is an extremely important process. The cell has to import all kinds of things that it needs from the outside, like oxygen, glucose, other sugars, irons, vitamins, etc. And then, of course, has to get rid of things like CO2, other waste products, as well as specialized things which that particular cell is involved in producing. Now, we have been studying for many years this group of transport proteins, which are very specialized, probably better to think of them as pumps. They have inside of the cell two parts of the protein which bind to a molecule called ATP, which is the basic energy currency of the cell. It binds to and chemically modifies that molecule. That generates the energy. You can think of this part of the protein as sort of the motor part of the pump. And then this part of the pump is what sits in the membrane and is involved for deciding what exact specific molecule will get moved, typically from this side to this side. And this is relevant because the gene causing PXE is one of these pumps. And so in general, our approach to genetic diseases is that we identify families with conditions that seem to be inherited. This is an extremely critical part of identifying these families, of characterizing them very carefully, which members of the family have the condition, which ones don't. Obviously, getting consent for them to participate in research studies collecting their samples, blood, DNA, etc. And from that process of studying the inheritance of genetic markers in those families, one can identify the gene involved. And this was the process that was taken with PXE, is that many families from around the world were collected and studied genetically, initially to find the chromosome where the gene lay, and then eventually to identify the gene itself. Our lab was not involved in that part of the process for PXE, although we were for a number of other genes involving ABC transporters, the first one being the gene for cystic fibrosis. Now, in principle, the whole idea of doing this is to identify the gene causing the disease, because from there you hopefully can learn what is the function of the disease, what is the normal function of that gene, and then how is that function disrupted or missing, causing the disease itself. And then the hope is from there, from having that knowledge, that one can go to a knowledge-based therapy. Many inherited genetic diseases, and PXE is certainly one of them, have quite complicated manifestations. There are a number of different symptoms, a number of different processes going on in the cell. Sometimes it's rather straightforward to figure out what the gene is and what's wrong and how you might fix it. Other times there's really no idea. And in PXE this was the case, a disease of elastic fibers in the skin and other parts of the body. And for literally decades people were looking at proteins involved in elastic fibers and other processes and getting nowhere. And in fact PXE was a surprise to be a transporter. And so there really was the only way to figure that out was through this genetics approach. Okay. Now, PXE is a genetic disorder. It's what we classify as a recessive genetic disease. That is, the people that are affected inherit two defective copies of the gene, two mutated copies, one from each parent. And so within a family, everybody who's affected, brothers and sisters, have to inherit the same two mutations, one from the mother and one from the father. So in this example, if we can distinguish, this would be the mother, this would be the father, if we can distinguish their two chromosomes, you can see that the three people who have the disease inherited the A chromosome from the mother and the D chromosome from the father. And this individual who's not affected by the disease inherited the non-disease or the normal chromosome for the father, happened to inherit the disease chromosome from the mother, and this individual would be a carrier. That is, if they married another person that was a carrier, they too could have children with this particular inherited condition, and that's how recessive genetic disorders operate. Now, virtually all of the genes that, like PXE, where there's a clear inherited basis and you can identify families with multiple affected individuals, we've basically solved genetically and identified the genes. So there's very little work to be done in those sort of families. There's still families out there where 2 or 3 individuals have a condition which looks like it's inherited, which are still being studied. So still very rare families with genetic conditions being studied, but all of the major genetic diseases have now at least been solved in the level of finding the gene. So what we're doing now is geneticists is mostly trying to tackle all the rest of the diseases. That is common complex diseases, things like diabetes, obesity, alcoholism, schizophrenia, most cancers, where we feel, and it's pretty clear, that every condition that you can think of is influenced by our genetic background. And so to identify those genes that are involved in susceptibility, we do studies where we collect individuals who have the condition in question, say a cancer, and those individuals from the population that don't have the disease, and then genetically compare the two. we can't use the family-based studies like we do in genetic diseases, because even though a disease like diabetes might be more prevalent in certain families, it's not inherited as a simple, clearly inherited genetic disorder. Same for obesity, same for alcoholism, same for schizophrenia. The reason your doctor will or should ask you your family history of what diseases are present in your family is that virtually all of these conditions can show clustering within families, but they're not strictly inherited. Okay, so then we separate basically two major types of diseases. Those that have a clear genetic inheritance we call Mendelian, in honor of Gregor Mendel, who was the individual who first basically developed genetics as a science. And so these would include not only PXE, but cystic fibrosis, Duchenne's muscular dystrophy, Tay-Sachs disease, sickle cell anemia, and hundreds of other diseases that are caused by alterations in a single gene, and their inheritance can be followed and tracked and predicted in a simple fashion, or at least in a straightforward fashion. Now, that does not mean there's not complications in all of these diseases. Many of these diseases, the severity of the disease can vary tremendously between families or within families. There's influence from the environment, etc. And we distinguish those from complex diseases, as we were talking about, cancer, asthma, schizophrenia, that show non-Mendelian inheritance. They may have multiple individuals in a family, but not inherited in a clear way. And we believe that's because these conditions are caused by multiple genes or genes and environmental interactions. Okay. So, like I said, where basically we're at with PXE is this situation. Years ago, we identified families with a condition. Many of those families very graciously volunteered to participate in a study where the gene was identified. Not too bad. So there we go. What I'm going to tell you about is another condition which, like PXE, is autosomally recessively inherited, where we've actually made considerably more progress and show you how that came about. And the disorder is called Stargardt macular dystrophy. These are patients who have degeneration of their macula, which is the center of their eye, and vision loss, starting in the age of young adults, teenagers to early adulthood. They have a characteristic degeneration of the retina and deposits in the epithelial cell behind the retina. As I said, this is autosomal recessive, relatively rare disease. About 1 in 10,000 individuals are born with this condition, but it's actually the most common genetic cause of blindness. So obviously it was important to identify the gene involved in this. And when we got involved in this work, families had been identified and a region of chromosome 1 had been indicated where the gene resided, but the actual gene had not been found. Now here's what the eye looks like in someone with Stargardt's disease. This is someone's normal eye, and you can see a very smooth appearance of the retina and a very even pigmentation. Here's an individual with Stargardt's disease, and you can obviously see these yellow deposits all around the eye. These are both lipids and retinoid vitamin A-like molecules that are depositing there abnormally. And then if you can see this very center part of the eye, it looks damaged or scarred, and in fact it is, and that represents basically death of the cells in the very center of the eye. Now you may think, well, this isn't a big portion of the eye that's showing that damage, but this is the macula, the center of the eye, where basically all your fine vision takes place. So when you're reading or watching television or even recognizing someone's face, you're basically using this part of your eye for your central vision. And if you lose vision in this part of your eye, it can be very debilitating in terms of lifestyle. In terms of not being able to read, not being able to drive a car, obviously most employment would be a problem, even watching television. And we're getting better at visual aids to help people read books or use the computer or other things, but obviously this is a devastating thing to happen, especially to a young adult, somebody of 15, 20, 25 years old, that previous to this had no problems with their vision. Now, while we were basically in the process of studying other genes that were part of this group of transporter family, one of the genes we studied we named ABCA4. There's basically 7 different subfamilies of ABC transporters, A to F. So this was the 4th gene we found in the A group. And we noticed that it had a funny pattern of expression. It was expressed only in the eye and the retina, and nowhere else in brain, heart, kidney, liver, lung, spleen was the gene expressed. And we mapped the gene to chromosome 1 right in the region where other investigators had mapped the gene for Stargardt disease. And since we had a gene expressed only in the eye and in the same region where there was a genetic disease involving the eye, we thought there might be a connection. And we called up one of the investigators involved in mapping the families, was a good friend of mine, Mark Leppert, who we had worked on with cystic fibrosis. He's out in the University of Utah. And I called him up and said, hey, I think we might have the gene causing Stargardt disease. And he said, wow, that would be fantastic. And he said, I'm collaborating with a group in Baylor. And I said I had received some useful comments from a group at Hopkins. And he said, well, let's all get together, pool the families we've collected over the years, and see if you're right. And it turns out we were right. We split up the work to study the gene, and it's a very large gene, as is the PXE gene, and we found mutations in most, about 70 or 80% of the families. And it's expected from the recessive inheritance that patients had 2 defective copies of the gene, 1 inherited from each patient. And those were mutations which changed amino acid all up and down the protein. Some were frame shift mutations where a few bases of DNA was inserted or was missing. Some affected the splice sites. These are the regions of the gene involved in stitching the initial messenger RNA together. And a few mutations created replaced an amino acid for basically a stop codon and would create a truncated protein at that position. But one thing we noticed was that all of the patients had at least one mutation that wouldn't completely disrupt the protein, that would just change one amino acid or take out a few amino acids and could be partially functional. And this was kind of puzzling to us because it didn't quite make sense. Usually in a recessive disease, you find many of the patients have mutations that knock out, basically disrupt entirely the function of both copies of the gene. Well, while we were pondering this, researchers in Spain contacted us and they said, well, we have a family, a very large family with 8 children with a form of retinitis pigmentosa that nobody's ever described before. And because this is such a family, in just one family, we were able to map the location of the disease gene, and it maps exactly on top of where the Stargardt gene lies. And we're wondering, could it possibly be the same gene, ABCA4, which is causing Stargardt disease? And I said, well, the easy way to find out, we'll send you all of the primers so you can scan and sequence the entire gene and figure that out. And they did, and they found that those individuals each inherited 2 defective copies of the same frame shift mutation, which basically caused the protein to be disrupted right at the front, completely knocking out the function of the gene in both copies. And so we came to the conclusion, well, if you did that, if you completely knocked out all function of ABCA4, you did not get Stargardt disease, but you got another eye disease, retinitis pigmentosa. Now, a year or 2 later, Frans Kramers in the Netherlands was studying families with cone rod dystrophy, another somewhat similar but clearly distinguishable eye condition. Again, he had recessive families, and he mapped some of them to the same region of chromosome 1, and he found again that those individuals had 2 defective copies of the very same gene, ABCA4. So now we had a situation, very exciting, where there were 3 different eye diseases that an ophthalmologist would characterize as distinct disorders based on their clinical appearance, all caused by mutations in the same gene. And we don't, in fact, still today don't have a really accurate way to measure the function of this protein ABCA4, but we could come up with a model if you were completely missing activity. You got a retinitis pigmentosa phenotype. If you had somewhat more activity, you got what a clinician would call cone rod dystrophy, and if you had a little bit more function of the gene, your phenotype would be Stargardt's disease. And so the phenotype depends on what we think the amount of function in the gene. And this points out that defects in one single gene can cause actually quite different phenotypic manifestations. In this case, it sort of is true within a family. If in a family one child has Stargardt's disease, other affected children in the family basically have the same condition, and the same with cone rod dystrophy, and the same with this rare family from Spain who had retinitis pigmentosa. But even within families or within different families with Stargardt's disease, we can find a lot of variation in terms of age of onset. Some of those children start to lose vision in 10, 15 years old. Others are in their 40s or 50s before vision loss starts to take place. And we don't really know why it might be so different in some families and not others. Now, in the case of Stargardt's disease, and we had a transporter that was in the eye, it was fairly straightforward in the next couple of years after this to figure out, make a very good model of what we think is going on. Now, this is a photoreceptor cell in your eye. In fact, they're just the very end of a rod photoreceptor cell, and it's made up of a large number of these discs. And inside these discs are basically the molecules involved in sensing light and then sending a signal to your brain indicating that you have seen light. And so that the major component of that is a protein called opsin, and inside that protein is a molecule of 11-cis-retinol, which is a form of vitamin A. Now that molecule is very sensitive to stimulation by light, and somehow I'm having trouble advancing the slide. Good question. Oop. There we go. Okay. I'm having some trouble advancing this slide, but basically when light sees this vitamin A-like molecule, it gets chemically converted from the cis form to a trans form, which is a slightly different shape of that vitamin A molecule. That releases the opsin protein, which can go on and then send a signal to the brain, to the nerve cells in the optic nerve that your eye has seen light. But then a whole complicated process has to happen to those vitamin A molecules. Vitamin A is one of the so-called fat-soluble vitamins. It does not dissolve in water. So it actually has to get physically linked to a fatty acid molecule in this membrane of this disc. That complex transported outside to the outside into the cell, an enzyme coming to release that vitamin A molecule, a transport process to remove that from the cell where it can get converted back to the cis form, transported back in, and go back and form more of this complex with the opsin protein. Now this is happening at an incredible rate. When you go out to bright sunlight, this is happening at a rate of 10,000 times per second. And so a lot of this molecule has to be generated. And what we believe now is that ABCA4 facilitates the movement of this lipid and vitamin A complex from the inside of this disc to the outside of that disc. And that's very important because these discs get recycled actually at an incredible rate of 10% a day. 10% of your rod photoreceptor cells get chewed up by these epithelial cells nearby so that these discs can be, all this component can be recycled so that you can have room to generate new discs which are all ready to detect light. And so if this vitamin A-like compound and this lipid are not removed from here, they will end up down here in these cells nearby and basically create garbage that shouldn't be there and that is toxic to these epithelial cells, poisoning them. And so that explains that damage that we see in the very center of the eye of someone with Stargardt's disease. So this was nice that now we could go from a disease where we didn't have any idea what the gene was involved to identifying the gene and protein, now to a pretty good model of what the function of that protein is and what's going on. It explains why the gene was expressed in the receptor, in the photoreceptor cells, but actually the damage is in the epithelial cells nearby. And that explains why, because these epithelial cells are accumulating these compounds which they shouldn't be, and so are thereby damaged. And this then could lead to a number of ideas of how you might fix this process. And if we can advance the slide, there we go. And so some other researchers hypothesized that the actual chemical that would be damaging would be an aldehyde, that this cis-retinol, which is an alcohol, get converted to an aldehyde, which is a molecule which is much more reactive, and large amounts of that molecule can cause all kinds of damage in those epithelial cells. Well, it turns out that conversion happens by an enzyme that we happen to have a drug that's sitting on the shelf of the pharmacy in your local drugstore that would inhibit that. And it's a drug called Accutane, or its chemical name is isotretinoin. And it's actually prescribed, one of the things it's prescribed for is to combat acne in people that have very severe problems with acne. And so those researchers hypothesized that maybe that compound would work in inhibiting the production of this aldehyde and might inhibit production of the damage. Well, meantime, other researchers had generated a model of Stargardt's disease in mice. They had disrupted the ABCA4 gene in mice and shown that it generated a condition in mice very similar to in humans, and they treated those mice with this Accutane and showed significantly less lesions and damage of the epithelial cells in the mice. And so this provided at least a proof of concept that this was probably the mechanism of action of damage, and that it would be possible pharmacologically to prevent the progression of this damage from happening. Now, there's a problem of giving Accutane. We actually can't give it to people who are adolescents and still growing because large quantities of the molecule that you would need in quantities enough to block this damage would be toxic. And so we're back to thinking of other ways that we could sort of chemically modify this process, but at least it provided us help that that this was the basis for the disease and that there would be some ways to intervene. Okay. Now,
could I interrupt for a moment?
Sure.
This is Christine Vokey. There were a couple attendees who joined a little bit late, so I just want to let people know how to ask questions during the presentation. There's a question pane on the right side of, in the control panel on the right side of your screen. You can type your questions into the pane, and the moderators, will see your question and present it. And we do have a question from a researcher in Philadelphia. And she asks if you can briefly explain how to deliver Accutane in vivo.
Well, normally we're given for Accutane. Accutane is taken orally. That would be a good question. We know that we can't give a high enough dose orally to get a high enough concentration in the eye, but it's not inconceivable that we could deliver Accutane or something like that in eye drops or even, as I'll talk in a minute, there's also subretinal injections that it's possible, and you may be familiar with this, of course, being used in PXE to fight some of the eye conditions that happen in PXE. So that's something certainly that's being considered, is could one develop a pharmacological agent in high enough concentration to the eye through, say, eye drops or an injection or a pump, rather than having to deliver the compound orally, where you have the problem, obviously, of maybe having to give a quite high dose. So that, that is a good question and something that's being looked into. Okay. Now with PXE, we have a similar kind of thing to those three eye diseases, in that there is quite a bit of variability in the manifestation of the disease. If any of you are either parents of PXE children and you have more than one child, you may have seen that the manifestations are quite different, or if you're an individual with PXE yourself and have brothers or sisters with a condition, you may have seen that, or obviously, if a physician or researcher treating patients, or families with PXE, you've probably seen this, that there's quite a bit of variability in the age of diagnosis, on the severity of the major complication, which gets identified first, the laxity in the skin, or the presence or absence of eye complications or heart disease. And so that even within the same family, where, as we discussed, with a recessive genetic disease, a brother and sister in the same family basically had to have inherited the same two defective genes. And so at the level of the PXE gene, they're identical. How can we explain that then they might have a different age of diagnosis or a different spectrum of symptoms? And so we're going to talk about that a bit, but one thing we think of as different from PXE is that the gene is actually somewhat removed from where the condition is manifesting itself. So with PXE, we're basically stuck at this part of the process. Like I said, we've collected families, we had localized the gene was identified, and now we're still really trying to figure out what is the exact function of that gene. We know we are almost completely sure that it has to be a transporter, because all of the genes related to it are transporters. But what is it transporting? What is its function? How does that exactly cause the manifestations of the disease? And without knowing that, we can't really design of an intelligent or a directed therapy without knowing what exactly is the problem. So as we've discussed, there are a number of different manifestations that occur in some patients, or not necessarily all patients, of loss of elasticity in the skin due to calcification and damage of elastic fibers in the skin, leading to this problem of skin laxity, to calcification in arterial vessels, which can lead to cardiovascular complications, to a particular manifestation in the eye of angioid streaks, leading to hemorrhages in the retina. Here's a case where there's actually been significant advancement in treating that most severe manifestation with actually medications which block the growth of new blood vessels, being found to be quite effective in stopping the progression of that damage and vision loss in PXE. And that's actually a medication developed to treat, originally developed to treat cancer, because we know tumors, as they grow, need a blood supply. And so the theory was, if we can inhibit the blood supply of a tumor, we can inhibit the growth of a tumor. It was later found that those agents actually work very effectively in retinal, in age-related macular degeneration, a very common condition involving overgrowth of blood vessels and bleeding of vessels in the eye. And from experience of that compound, the process was extended to individuals with PXE and found to be quite effective. Now the transporter that we call ABCC6 that causes PXE is mostly found in the liver and in the kidney, but as you can see, these manifestations are out in the skin, in the blood vessels, in the eye, in the heart, and so we're really at a loss to explain why does this transporter in the kidney and the liver have these manifestations out in other organs. And so here is the human liver, which, as you may know, takes in blood both directly from an artery, directly from the heart, and from a vein from the intestines. And that blood flows into the liver and has a number of functions, mostly to filter out compounds that shouldn't be in the liver. Some of that filtering goes into forming the fluid in the bile, which passes into the intestine, is very important to transporting cholesterol and other compounds. And the blood leaving the liver goes into the hepatic vein back to the heart, where it gets pumped out again. So one of the major functions of the liver is as a filter to filter our blood, but the liver is also involved in producing a number of compounds which get transported through the blood around to the rest of the body. And we know the ABCC6 transporter is not in the part of the liver transporting compounds into the bile. There are actually a number of other ABC transporters that are there and are involved in doing that, but is out here transporting compounds which go back into the blood. So our speculation, which we think virtually everybody agrees on that has studied that, is that the ABC transporter is sitting here in the liver transporting some compound into the blood to go back and circulate across the whole body. So that could explain why a defect, why missing a transporter here could have effects all across the body if that transporter was transporting something critical to different cells across the body. And so we have evidence then that ABCC6 transports a critical molecule into the blood, controlling calcification in the elastic tissue and throughout the body. And this is, at the molecular level, what this calcification and damage to elastic fibers looks like when you do a biopsy of the skin of somebody with PXE. But to date, all efforts have not been successful in trying to identify what is this mystery molecule causing PXE. So let me go through a few of the things that have been tried, explain why we took some of these approaches. And these are actually two approaches that we worked on in my laboratory. One was identifying an animal model. If we could have an animal model, this whole process, maybe in that animal model we could use to figure out what this mystery compound is. And then the other approach we've taken was basically a chemical approach, assuming that this might be a very small molecule and using very advanced new chemical technologies like nuclear magnetic resonance and mass spectroscopy to actually identify the compounds directly. So the model system that my laboratory was interested in developing was zebrafish. Now I used to have pet mice, so I don't particularly like working with mice as a researcher, but was attracted to the idea of using these fish. Now fish are a vertebrate just like us. They have eyes, they have skin, they have blood, a heart. Mostly all of the organs and bones and systems that we have are present in the fish. And often remarkably similar to that in the human body. Now it's possible to do all kinds of very sophisticated genetic experiments with these fish. You can breed hundreds or thousands of them very rapidly and look for new mutations or even screen for particular conditions that you might want to find. When we started this process, the genome sequence of the zebrafish was just being assembled, and the genomes of two other fish had also been characterized, two pufferfish. They were chosen because their actual genome is quite small, and so sequencing them and assembling them was fairly straightforward. Now there are 48 of these ABC transporter genes in the human, and when we looked in the fish, we found that 32 of those were conserved, and we had good evidence that the fish version of the gene was evolutionarily related to or the same as the human version of the gene, and so might be a good model for studying the function. And in fact, we have 10 or 15 human ABC transporters that still to date, we don't know what their function is in the human. So we thought, well, if we can figure that out in the fish and transfer that knowledge to the human, then we'll be further along. In the fish, they're actually completely transparent in the early stages as an embryo, and so if you can label the product of a gene, you can basically light up the tissues in the fish where that gene is expressed and get some idea of where the gene's expressed and what its function might be. And then there's another technology where you can make an oligonucleotide, a small piece of DNA, that will specifically target any gene and inhibit it or suppress the production of that protein. And you can inject that oligonucleotide into a fertilized egg of the fish, and then that protein will be suppressed as the fish develops, and you can see what is the effect of doing that. And so it's a quite inexpensive and rapid way to figure out the function of the gene. And so when we applied this then to ABCC6, the PXE gene, we found that the zebrafish in fact had an ABCC6 gene that was quite similar to human and mammalian and other vertebrate genes. This is a gene tree where we basically take the amino acid sequence and basically compare it to see which ones are more like each other. And the rat ABCC6 gene and the mouse ABCC6 gene were the two closest to each other, and that made sense because rat and mice are close to each other. This is the human gene, and the rodent genes were closest to the human gene. And here's the chicken gene, and it was the next closest. And here's the fish gene, both zebrafish and then another related fish species. And those were all closer to each other than any of the other genes in the C class of ABC transporters, either in humans or in zebrafish. And so this provided to us quite convincing evidence that this gene, which we had named ABCC6 in the fish, was really the fish version of the human gene responsible for PXE. Now the story got a little more interesting. From further digging, we found that there were actually two ABCC6 genes in the fish, and we labeled them A and B. And a group in Europe actually did the experiment of labeling them and looking in the fish at where they expressed. And one of them is expressed in early development in cells which go on to become the liver. Another is expressed in the cells in the embryo which go on to form the kidney. So this is quite interesting. In the human, we have one gene expressed in both the liver and the kidney. In the fish, we have two genes, one in the liver, one in the kidney. And they also performed this suppression experiment. When we suppress the 6B gene, the one in the kidney, we couldn't see anything happen. Those fish looked to us completely normal. Now, you can only follow this out several days of development when the fish have hatched and have started swimming around, and most of the body is intact, but you can't really then grow the fish up to adulthood and study all of the functions. So we couldn't really figure out, does the kidney develop completely normally and function completely normally in the adult if you were to suppress this protein. But the 6A version, the one expressed in the liver, had very severe consequences. Those fish embryos, their heart basically expanded with fluid, and they developed very abnormally, and those that developed that condition died within 2 or 3 days of development. So obviously that gene has something very important to do in the liver, again causing effects outside of the liver in other parts of the body, in particular in the heart. Now that's sort of where we've ended up, and the story hasn't progressed further. There still could be quite a good animal model to look at PXE, I think. And if if I had a wish list, if Warren Buffett came to us and said, "Here's a blank check, solve PXE," One of the things I would definitely say was that we would put a whole team together studying these fish and maybe make a real mutation in the germline of the fish so we could grow them up to adulthood and figure out in more detail exactly what's going on. But so far we don't have that blank check to make that happen. Now, meanwhile, at least two groups had made mouse models of PXE. They had knocked out the gene for ABCC6. And what has been fortunate, mouse models for human genetic diseases don't often show the same phenotype, but in careful looking at regions of the skin of these mice, you can see a calcification defect very similar to what happens in the case of the human. And curiously enough, there had been mice identified decades ago that had calcifications of their heart, and when those were looked at in detail, they were found to have mutations in ABCC6. So we do have a mouse model of the disease, and that obviously is being looked at and will be a very important thing to potentially test new therapies in. Okay. So all of these studies have supported what is being known as a metabolic model of PXE. That is, some substance, as we discussed, some substance from the liver is probably missing or potentially could be overabundant. But since we think the function of the gene is missing and it's a transporter transporting into the blood, we like to think that the molecule is missing, causing this mineralization out in the skin and the eyes and the heart and all over the rest of the body. And this is actually supported by some other experiments. There have been some interesting cases where an individual needed a liver transplant, received a liver that happened to be from an individual that had PXE, that didn't know it at the time that person died. They died of some other condition and their liver was donated. And so it seems to me, it seems from this evidence that if you move a liver from a PXE patient into someone who doesn't have PXE, that new individual basically generates PXE. So basically validating this model that the cause of the disorder is a defect in the liver, and it's something metabolic, it's something circulating in our blood. As well, Olivier Lassault in the University of Hawaii has done some experiments taking blood and serum from individuals who have PXE, putting them on normal skin cells, and show that they develop a calcification-like phenotype in culture, basically replicating the whole process of PXE in a dish. And so this is giving us some hope that we might be able to use that process to actually purify the molecule that's causing that, or the molecule that's basically missing. So a study that we attempted, and that some of you who are watching and listening may have actually participated in, was to directly try to study, in this case, the urine of individuals with PXE. We reasoned, since PXE is also expressed in the kidney, that it might be also transporting this mystery compound into the urine. We chose men because women, because of all of the hormones and hormone metabolites they have, the urine of women is more chemically complex and harder to study than men. And so we had plenty of men who were willing to volunteer to give a sample of urine, which we collected, and we collected from control men who did not have PXE, and using current technology, tried to look for a molecule in the urine, PXE men, or missing in the urine in PXE men, which was present in men that didn't have PXE. It turned out there were just too many compounds, that the complexity, chemical complexity, was too complex for current technology to figure that out. So once the mouse model was derived, we turned to the mouse model and basically did similar experiments in the mouse. And if this will advance, this shows here the urine from mice with PXE, and urine from mice that don't have PXE. And in the purple are spots of chemicals that are present over here that are not over here. And in green are chemicals that are present, or at least a higher amount over here than over here. But you can see it's still quite complicated. It's not like there's just one spot present on one side and not to the other that we could point to and say this is the molecule. Plus, there's no really good map for what all these chemicals are. To figure out what they are, the technology does give us the molecular weight of the compound, and it is possible to go one at a time and basically purify this spot and blast the chemical apart more and figure out exactly what it's made of. That can be done, and we've done some of that, but we've not come up with anything that was obvious and that made a lot of sense. But again, I think this approach would still work. So again, I think if somebody knows Warren Buffett or some other billionaire willing to write a very large check to study PXE, this approach could be advanced further, and we think would generate useful information. Obviously, since we think the missing molecule may be in the blood, looking at the serum of PXE in normal mice is something else that we've certainly considered. But as I said, the resultant pattern is too complex for us really to figure out what the mystery substance is. So let me talk then in general some of the concepts that have been advanced for correcting genetic diseases or treating genetic diseases. So the most obvious would be gene therapy. If we could just replace a defective gene in all of the cells in an individual with a genetic disease, then in principle that would cure the disease. That turns out to be not practical to do in most cases. It has been tried in some blood disorders where there's a gene missing in cells of the blood, and one can take out stem cells from the blood of those children, put in the defective gene, and put it back again. And you may have seen reports of children with severe immune deficiencies, sort of so-called boy in the bubble, these kids that basically have no immune system and can't be exposed to any germs from the outside. Those experiments were tried there with some success, but the problem what happened was introducing a new gene into cells of the blood can potentially disrupt other genes and in fact cause cancer, and that actually happened in some cases. And so obviously we don't want to trade one disease for another and cure the genetic disease, but result in cancer. Now, organ replacement could be a possibility, and this is not a completely far-fetched idea for PXE. And in fact, we think if you would give a liver transplant to someone with PXE, you might in fact cure the condition. Now, obviously, the PXE is not a severe enough condition in most individuals that one would go through either the expense or the complications of liver transplant. Liver transplant involves irradiating yourself and making your body immune suppressed in order that you don't reject the donor organ. So that's probably not a practical practical solution. But one thing interesting about the liver is it does regenerate it, so it's not inconceivable that down the road we won't be necessarily transplanting the liver, but we might actually be able to replace or remove part of the liver and have it regenerate with a liver that had intact PXE gene. So that's that's something that's not inconceivable. And then stem cell therapy is another idea, or a variant of that. Could we generate liver stem cells which we could introduce that would put back, if you will, this mystery molecule. And maybe we don't have to replace all of the liver. Maybe you only have to replace 1% of the cells in the liver to get enough of this compound to reverse progression of the disease. Now, obviously for a metabolic disorder, we would think that maybe there would be some medication, maybe there would be some pill that we could give that would replace the missing molecule or replace the need for it or prevent the calcification, for example, to happening. And we're actually quite hopeful that that could be the case once we figure out what this mystery compound is. We've had some interesting leads over the last couple of years that it might be vitamin K, and that was advanced for a long time, and we're very excited about that. That turns out that the CEC6 is not a transporter of vitamin K, as far as we can tell. But again, if we could figure out what the mystery substance is, that we might be able to think of some way to be able to correct that. That wouldn't be a cure necessarily, but if it prevented the progression of the disease and was a medication without any side effects, that we could conceive of treating an individual with PXE through most of their lifetime. Now, there are some genetic conditions where, if recognized early, we can manipulate the environment of the individual to prevent the manifestations of the disease. And the most famous is PKU, or phenylketonuria. You may be familiar with any baby that's born in the U.S. or most countries has a spot of blood taken off of the bottom of their foot, and that's used to test for this condition. It's a condition which causes mental retardation. It's a defect in metabolizing phenylalanine, which is one of the amino acids that we have in the proteins of our body. And that mental retardation is particularly susceptible to happen from the period after birth until the first couple years of life. So what can be done with these children if they're recognized early or at birth, basically while they're still nursing and before they're given any foods which have phenylalanine, is they can be given a special diet, low in phenylalanine, and the dangerous compound which causes the damage to development of the brain is not present, and so the child develops their brain normally and then can basically have more or less a normal lifespan. In PXE, we don't think that is an issue, that typically manifestations are happening in adolescence and beyond, but a similar kind of thing, that maybe we could change the diet of individuals to prevent the manifestation of PXE. We think that's probably not the case, because we think that there's something missing in the blood which needs to be replaced. Now, I just happened to open my newspaper this morning and saw an article on the front page of the Washington Post how stem cell therapy had cured or had improved the eyesight of 2 patients with eye disease. And as I read down further, I saw that one of those was a patient with Stargardt's disease, the gene that we had identified years ago. And what they had done is basically taken stem cells from normal individuals and regenerated in culture those epithelial cells found in the retina, cultured enough of those cells, and injected them into the eye, in this case into one eye of an individual with Stargardt's disease. And lo and behold, 12 weeks later, that person is now starting to see. Both by visual acuity, they went to a visual acuity where basically all they could see was a hand waving in front of them, to be able to count fingers on a hand, to now having a measurable visual acuity, and to be able to see on an eye chart no letters to go from 1 to 3 to 5. So this is obviously very encouraging. It's only a single patient with this condition and a single patient with age-related macular degeneration. But obviously, this is generating a tremendous amount of excitement, both in the ophthalmology community, obviously in the individuals and families with both Stargardt's disease as well as macular degeneration, which is a condition that affects 10% of all Americans and is the #1 cause of blindness in the elderly in the United States. So I think we're obviously going to hear a lot more about stem cell therapy for certain conditions. And now we have the possibility of not needing to generate stem cells from a fertilized embryo, which obviously has ethical implications, which some groups object to. But now we can actually generate stem cells from the skin cells of virtually anybody, basically induce those cells to go back to being early embryonic cells, where one could generate cells from the eye, cells from the kidneys, cells from the liver, and potentially introduce those cells back in the body. I think we're quite a ways from attempting that with PXE, but obviously having a mouse model and even a fish model may potentially help us in developing and in improving those therapies. Okay. Last year I'd like to talk about a therapeutic approach actually that we're applying to cancer. I didn't say this in the beginning, but one of the reasons we're studying these AVC transporters, these pumps, is they're incredibly important in cancer. Many tumor cells overexpress these pumps and use them to pump out the same chemotherapy drugs that we're giving to try to kill cancer patients. And this is a major reason why many tumors develop resistance to chemotherapy and then become untreatable. So in the case of cancer and the pumps that are pumping chemotherapy drugs, we thought of ways to try to inhibit the pump. And the pumps are composed of clusters of chains of the protein, which form these sort of spiral helical structures which go across the membrane. And they're formed of amino acids that basically are neutral in charge, and so they will tend to cluster together in this clump and are essentially like fats. The membrane is made up of mostly fats as opposed to the liquid surface which is on either side. So a chemist in our institute, Nadia Tarasova, decided to synthetically make one of these loops, put a charged molecule on the outside so that we could get it, in fact, into solution and deliver it to cells. And lo and behold, these became very, very sensitive and selective inhibitors for these pumps that are pumping chemotherapy drugs. And what we think happens is those loops go into the membrane, into the protein where they're supposed to be, and basically kick out or disrupt the normal part of the protein which is supposed to be in there. And so then become specific inhibitors that way. This could be relative to PXE, is that we could use this as a very specific inhibitor to ABCC6 and use this as a research tool to inhibit just that transporter and maybe figure out in a cell-based model what the compound is pumping, what the transporter is pumping. Curiously, and I show this just because it just shows how you never know what to expect out of research, when we dissolve those little loop-like compounds, these compounds in water, we found that they form spontaneously all by themselves into little spheres. And that kind of makes sense because they don't like being near water, and so they tend to all clump up together to themselves, but they form these very nice, even-looking spheres. And here's a measurement of the size of them. They're virtually all the exact same size. Now, when we showed this to a biotech company, they said, wow, that's very interesting. I wonder if we could use these spheres as kind of like nanoparticles. And then we showed them another data where we had taken a chemotherapy drug which doesn't dissolve at all in water and threw in these little spheres, and lo and behold, now the compound, which is yellow, goes inside the center of those spheres and now basically can be carried in this aqueous solution. And so now the biotech company we've been talking to is now even more exciting that these nanospheres could be used to deliver compounds that you can't normally get dissolved into blood and deliver them out to tissues or maybe out into blood vessels where they might be needed. And this could be useful not only for delivering compounds to tumors to try to kill them, but maybe to deliver other kind of compounds, maybe even the mysterious mystery substance PXE, which might actually be a compound which doesn't like to be in water, but needs to be in a special, needs some other transporter to transport it through the blood. So this is just an example of you sort of never know in research where you're being led. And I actually got involved in this because I was studying cancer, and a cancer gene we were studying called MET happened to be near the gene for cystic fibrosis. And we went on to find the gene for cystic fibrosis, which is an ABC transporter, which led us back to other ABC transporters involved in cancer, but also led us to study diseases like Stargardt disease that I talked about, or also PXE. So in conclusion then, there are really many routes to discover the genes that cause human diseases. As I said, we've solved virtually all of the cleanly inherited, obviously inherited human genetic diseases, and are now working on all of the other complex diseases that we find. So uncovering the function of the gene can sometimes be fairly straightforward, like it was for ABCA4 and Stargardt's disease. That only took a couple of years to figure out and to even come up with a reasonable idea of a therapy. At other times it can be extremely challenging, and PXE, I have to say, has to be one of the more challenging that have been come across. As I said, virtually all the genes for genetic diseases have been identified, and in the vast majority we have some reasonable idea of why that gene is causing the disease. In PXE, this is still something that's lacking. But we're quite hopeful that once we figure that out, that the pathway to go from there to a treatment or a therapy could happen quite rapidly. So there's no easy approach to replacing a defective gene into an adult human, especially a gene that has actions all over the body. But as you see, we're getting closer and closer to the day we can use organ transplant, where we can use stem cells, we can use cells derived in culture and generate them back into individuals. And so this will be, in the future going forward, something that will be certainly tried more and more. So therapies and treatments, at least for PXE, to date have really had to rely on either avoiding the adverse effects, which we don't really understand what those are in PXE, or correcting the problem or problems with medications. And obviously we can perform cosmetic surgery on people with bad skin laxity and correct that in that way. As we discussed, there are medications for the retinal hemorrhages in the eye, which are showing considerable benefit. So that's sort of treating their symptoms and treating the manifestations as you go along, which is not the ideal. We would much rather prefer to be preventing the progression of the disease. And I threw this in at the end because this came up earlier. So new gene sequencing technology is making all of this approach happen faster. And I actually just got back from a trip from China where I saw in single room more DNA sequencers than exist in any other institute in the world. And so the technology now actually to sequence all of the genes in an individual can be done for approximately $1,000 and in as quickly as a day or two. So that for genetic conditions like PXE, or that are difficult to diagnose, like not everybody that receives the initial diagnosis from PXE is that certain. And in fact, there was just a publication showing that there may be a second gene that causes some similar manifestations to PXE. So now we can conceive of using this new gene sequencing technology to at least tell us exactly yes or no. Are there defects in ABCC6 in this individual with skin laxity and calcification of their skin that we're not sure of the diagnosis of, or might it be another gene, and might separating those individuals more precisely genetically allow us better able to study the conditions. So much progress has been made in lots of genetic diseases and in understanding how genetic diseases work, and to some extent in PXE, but we're still missing this key part of the puzzle. And I'm sure this is frustrating for the patients and their families and their clinicians, and it's equally frustrating for the clinicians because we like to solve problems and we don't like when problems take many, many years to solve. And certainly PXE International has been very, very instrumental in getting us to progress where we are this far. It's difficult to get the government to put large amounts of funding into rare genetic disorders, and PXE International has been one of the leaders in really showing that this is a worthwhile thing to do. We're learning very interesting things in studying PXE that might have ramifications for cancer, for eye disease, for heart disease, for skin diseases, and so it's really to everyone's benefit to find this missing puzzle piece and solve this mystery, because every piece of the puzzle of how the human body works and how it doesn't work properly in disease is useful to advancing to the day when we can eliminate disease. So I'd like to stop there and thank you. Here is more information and contact about PXE International, about these webinars and where this webinar will be stored after this is done, and about upcoming webinars that you may also be interested in. Thank you very much.
Thank you very much, Dr. Dean. We do have a couple questions. One is the role of magnesium and some of the results in the PXE mouse. Can you explain in any way or address what that might be doing biochemically, and maybe talk a little bit about the clinical trial that will be taking place hopefully this year in humans?
Yeah, I'm not as directly familiar with that part of the process. I mean, magnesium is a mineral much like calcium, and we know that they function together or in opposite ways in a lot of different parts of the body. So there has been thought that by by manipulating magnesium, we might be able to manipulate or prevent or this process of calcification. But yeah, I'm sorry, that's not really an area of my direct expertise.
Okay, thank you. And then we have a couple questions from researchers. Do patients with PXE have abnormal blood glucose levels?
I think that has never been found consistently. So I think not. Now there may be, obviously there may be some manifestations of the disease process happens, but no, I'm not aware that there's a consistent blood glucose abnormality.
And one more question from a researcher about the mouse urine samples that you analyzed. Have you analyzed or thought about analyzing mouse serum samples using the same techniques?
Yes, we've definitely thought about that, and some of that has been tried. I didn't show the data. Serum we started with urine because it's, at least in terms of its spectrum of small molecules, it's much, much more simple than serum or plasma, which has many, many more small molecules. But yes, we have, we have tried that and also seen it's very, very complicated, and there are a lot of spots present in the PXE mouse that are not present in the normal mouse, and vice versa. So it's been too hard to interpret. Wynn Arias actually had a brilliant idea, says that if we could change the ABC6 protein and get it to target to the part of the liver which goes into the bile is actually chemically quite simple, and maybe we could trick the liver of a mouse to transport the mystery compound into the bile, collect the bile, and then study that. So that was actually an idea that came out of our ABC transporter meeting a couple years ago, which is a good idea. I don't know that it's been followed up on, but it's something that would be technically, technically feasible.
Well, very interesting. Well, I don't have any further questions, and I want to thank you very much for your time in explaining all this to us and for your involvement in PXE research. We are delighted that you're on this team. I want to also point out that the upcoming webinars in this spring, PXE and Your Child, will be happening on February 1st. You can go to the website for the exact times or to register. And that's Dr. Olivier Van Acker from Belgium who will be presenting that. And the 2012 PXE Research Update will be hosted by Patrick Terry on March 14th. Again, the times will be available on the website and registration. This webinar will be viewable as a video from our website within a few weeks. We'll probably send out an announcement that it's up there when it's available. And one more thing that's not on the slide, we have our biennial member meeting this year, September 21st to 23rd, 2012, in Bethesda, Maryland, the same location as before. So we hope many of you can make it. And again, Dr. Dean, thank you very much for your time today, and that concludes our webinar.
Thank you very much, and good day to all.